Hello everyone, in today's presentation I'll be talking about superantigens. We all know about antigens, we have heard about them one or the other time in our lives, but I'll be talking about superantigens, what makes them super. Before that, I'll be talking about acquired immunity because it is immunity that we gain during our lifetime due to interaction with the foreign particles that are antigens. They come into our body, activate many cells in our body, make our body produce antibodies. There is a phagocytosis that occurs when a bacterium comes in, its antigen is identified, antigen presenting cell, and being presented on MHC class 2 molecule that is kind of a billboard for another T cells that are roaming in our body to see that there is antigen in our body. It is being presented over this cell, so now come and act over it. This is how our acquired immunity mainly works. It shows this to the other T cells and they get activated. These T cells further activate and helper T cells or other cells. This is a complicated diagram, but I'll try to simplify it. In here, antigen presenting cell presents the antigen and it activates the T cell. Not all T cells are activated. Just the particular T cell which is supposed to be activated will be activated, and it produces intraleukins. These are just a fancy word for signals. These signals further produce T helper cells, activate T helper cells, and B cells, they produce antibodies against that antigen. This is how our immune system acts when some antigen comes. But what is a super antigen? A normal antigen is supposed to activate just few T cells, that is just 0.001 to 0.01%. That is a specific activation. These T cells just are specifically activated for that antigen. But when super antigen comes in, it just activates many T cells, like 20 to 30% of T cells. It is non-specific activation. This activation leads to the production of many induction of many T cells and non-specific activation. Non-specific and polyclonal activation. The polyclonal activation is just where these T cells don't know what they're supposed to do because they are activated, but in monoclonal, the specific activation leads to the production of many T cells, activation of many T cells. These act over one foreign entity, one antigen, but in polyclonal they are activated. It's just a way of super antigens to invade our immune system. This is their defense system against our immune system. In this, T cells don't know what they're supposed to do. They don't know which antigen they're supposed to act on. They don't have particular receptor site for all of them. How do they do this? Because not all T cell are no T cell can be activated by two T cells, but they just do not bind to the main site of T cell receptor. A normal antigen is processed by antigen presenting cell and it is binded with T cell, and T cell is activated. But a super antigen do not bind in there. It has a different shape. It binds on the outside and activate T cells. This way they can activate many T cells. So different T cells are activated and they do not have specific action against this super antigens. So there are just many T cells in our body and they don't know what they're supposed to do. There's just a chaotic situation. So they don't bind anywhere. They just bind on the outside. The different diseases caused by this are like staphylococcal food poisoning caused by staphylococcal enterotoxin. Then there's another staphylococcal TSST that causes toxic shock syndrome, and then rabies virus. In this, the superantigens help promote viral transmission. Cytomegalovirus associated superantigen enhance HIV replication. This has been observed that they enhance HIV1 replication in AIDS patient. This is actually herpes virus, but it's superantigen enhanced replication. How? Because it produces many T cells. So those T cells help. Those T cells are the main sites where HIV1 replicates. So this just helps in the replication of HIV virus in AIDS patients. This was one of the observations. So these are the different diseases which are being caused by which superantigens have been observed. So different drugs that can be used are cyclosporine, and there is a Chinese herb picoline which is used in cirrhosis, and receptor antagonists, genetically engineered proteins that interfere with the binding of superantigen to TCR. This can help in superantigen associated diseases. So superantigens in medical therapy, this is like a positive part of superantigens. They have been used in cancer immunotherapy and the treatment of infectious and autoimmune diseases. So their capacity to produce many antigens has been used. So conclusion, their quest has only just begun yet, because there are many microbes which can be bearing such quality, but we have not been able to distinguish them yet. So significant new insights into the pathogenesis of microbial diseases can be gained in the future by the analysis of this new type of antigen. There is still ongoing research in it, so there are not many microbes being identified with such quality with superantigens, but it can actually help to understand it better. Thank you very much for your patience.